Everybody eat lunch?
Oh, no.
Yeah. No!
We had breakfast.
That's
Nope.
not a good thing. That's a breakfast.
I'm getting
We had breakfast.
A good healthy breakfast. Cupcakes?
No. Pears.
Yeah,
Pears.
Sausage.
No.
Egg.
We
Sandwich.
had an ice cream.
You bought fast food.
No.
No? You cooked a sausage? You have. All right, energy systems. Okay, so we've looked at which food substrates we're going to use to make energy for exercise. And we looked at an ATP molecule briefly, and we looked at the idea of enzymes. So now let's jump into the systems. So the first system is the ATP-PC system. It's the only one of the three that does not use a food substrate to make ATP. All right, because any food that we have, if we're going to break down, even if it's a glucose molecule, that takes time. And there are times when we have to be able to contract muscles where we don't have time to wait to break down a glucose molecule. So we have to have a system that can work immediately. Right? So the ATP-PC system fulfills that role. A lot of energy in a very, very short amount of time. So sprinting, if I suddenly start sprinting, I have to have the energy to contract muscles to do that. All right, to stand up, as we did on Wednesday, took energy that you didn't have produced at that time in order to stand up. Lifting a heavy weight, so if I've got to pick a big box up. The ATP-PC system is very short term, so it doesn't last for very long, but it lasts long enough for the systems that break down a food group to then be able to take over. So what we rely on here is we have some ATP already stored, so I don't have to make it. I've got a couple of seconds worth sitting in the muscle. About, for some people, up to about 5 seconds. Right? So very, very little. I can't get very far in 5 seconds. So then I have to start making it. PC stands for phosphocreatine. PC is easier, right? So you can always look up the phosphocreatine. If you are hunting around online, it used to be called CP. It used to be creatine phosphate. And then somewhere between my undergrad and my graduate program, they flipped those two around and made it phosphocreatine, and I don't know why, and I haven't been interested enough to find out. So when you look and you see things about CP, they're talking about the same molecule and the same system, but it's a little out of date because none of the textbooks, none of the research calls it creatine phosphate anymore. So phosphocreatine is made up of creatine and an inorganic phosphate. And if you remember on Wednesday, we looked at ATP is an adenosine plus 3 inorganic phosphates. So I have my adenosine, and then I have an inorganic phosphate, an inorganic phosphate, and then I've got that energy bond I can get at to my third inorganic phosphate. Where? Oh, yes, adenosine, yeah. No, I was just trying to, yeah. Right? And remember, it's only this third energy bond we can steal for muscle contraction. Right? So once I've taken that energy and used it for muscle contraction, I'm left with a DP and free phosphate. Now, creatine phosphate is a creatine plus an inorganic phosphate. And so what I can do is I can break this down and I can steal the energy bond to fix the inorganic phosphate back to the ADP. Okay. So this energy bond in creatine phosphate is similar to the one that we see in the ADP. It's there, but we can't use it directly for muscle contraction. Right? I can steal it and make ATP again. And then I can break the ATP down and take the energy bond and use it. It's a little convoluted, right? Surely it would be much, much easier if we could just break the BC apart and take that energy for muscle contraction directly, right? But we can't. So we just have to learn that extra step in the loop. Yes, makes sense. All right, so our enzymes then, when we look at this system, we have an enzyme for breaking down ATP, and it's called ATPase. Anytime you see a word with ASE on it, it's going to be an enzyme. This particular one is easy because it's named exactly what it does, right? ATPase facilitates breaking down ATP and releasing energy. So it's very straightforward. The other one for this system is also straightforward. It's called creatine kinase. And creatine kinase facilitates breaking down phosphocreatine and releasing the energy bond to rebuild ATP. Right? Remember that those enzymes are very specific. Creatine kinase can't break down ATP. ATPase can't break down phosphocreatine. So they only do their one job. They're highly specific. They're very temperature sensitive. They're very pH sensitive. So, if I go all out, as hard as I can go, absolutely 100% run as fast as I can run, or I'm doing a 1 rep max, then at maximum intensity, both of these molecules could be used up in as little as 4 seconds. Right? Most of us don't do a lot of 100% max intensity stuff. We do 97% or 98%. So we have a little bit longer than 4 seconds most of the time. So energy system 1, ATP, we have about 5 seconds. If I'm breaking down phosphocreatine, on the whole, I've got enough phosphocreatine stored in my muscles for somewhere between 10 to 15 seconds, depending which textbook you're looking at and which individuals you're testing. So all in all, we've got somewhere between 15 to 20 seconds. If we're really, really lucky, we might be close to 25 seconds. But most people, it's more around 20 seconds worth of energy that we can make from that system. When I make the ATP and I break the ATP down, an inorganic phosphate is acidic. And we release some hydrogen, which is acidic. So one of the reasons we can't keep up that intensity for very long is even if we had more substance, the pH starts to get too high and the enzymes slow down, and so I can't make energy to maintain that force production. Right. So 15 to 20 seconds doesn't give us a lot of time, right? And we're out of substrate now. We don't have any ATP left, we don't have any phosphocreatine left, but there's still a tiger chasing us, so what do? If I'm training an event that is very short, very powerful, very strong, then what do I see happen with this system? Because one of the things that is interesting about exercise is our body adapts so that we become better at the thing we're practicing, right? It's not just my skill level that gets better with practice. I get stronger, I get fitter, I get more flexible. I see adaptation in my body in response to training. Right? So we can see obvious things that we can actually observe from the outside, but inside, in the cells, we also see adaptation when we train. So if I stress a system, and this is a system, it's just not one I can see from outside, right? If I stress a system, in response to the system will adapt to deal with the stress. So if I am a sprinter, and I do a lot of sprinting training, then I expect to see something change within the energy system to support that training. Otherwise, what would be the point, right? If I go down the gym and I lift weights every day, day after day after day after day, and nothing happened, why would I keep going? Right? There would be no point in training if we don't adapt to training. So this adapts as well. In people who are ATP, PC system dominant, so sprinters, jumpers, throwers, lifters, right? Then what we see is a change in the activity level, particularly of creatine kinase. So the faster, the more active the enzyme is, the more energy, the quicker I can get at the energy that's stored in the creatine phosphate. We also see this pathway start to work a little more quickly. So I can break the phosphocreatine down faster, and once I've got that energy, I can fix the ADP, add the PI, and make it ATP faster. Right? So people who are trained sprinters, you see something physiologically more advanced than you do in people who aren't trained sprinters. So if I can do that, if I can break it down, get at the energy and use the energy quicker, then we see increased performance in sprint events, throw events, jump events, for people who train those events versus the rest of us. So it's not just that they got stronger, it's not just that their technique is better, it's also that their energy system is working more efficiently and more effectively. Sometimes what we see is that my initial stored amount of ATP and PC sitting in the muscle cell goes up. And later in the semester, we'll talk about creatine as a supplement. because that's one of the reasons why we take creatine as a supplement, right? If I can increase the creatine level, maybe I can increase the creatine phosphate level, phosphocreatine level, and I can produce energy for 23 seconds instead of 20 seconds. But that 3 seconds gives me a big advantage. If I'm doing a lot of running, biking, walking, we don't see those adaptations because I don't use this system to provide energy for those kinds of activities. So if I'm doing a lot of running, then I wouldn't expect to see the adaptation that I want for a power event. So that's another reason. If I have power athletes, I don't want them doing heaps and heaps and heaps of endurance work because it's training the wrong system. There's only so many hours in a day. I can't have them working on their sprinting and running 5 miles a day and get the same effect, run a mile, run 2 miles, whatever you decide, right? But if I have them doing a lot of endurance work, then it's not going to improve the energy system that I need them to be training for their event. Right? Makes sense. Adaptation is specific as well. Right? I only adapt to the thing that I'm stressing the system with. So it's a little unclear how much we have to increase the resting levels of ATP and PC to see improved performance. So the whole creatine supplementation doesn't do any harm that we know of so far for most people, but it may or may not be necessary to enhance performance. But it probably will help you train a little bit harder. And if you can train harder, you should see an improvement in performance. So it could be an indirect pathway there. Questions on System 1? Who thinks they're a System 1 athlete? You died? Okay, no one, huh? All right. Okay, System 2. Did we use any oxygen in our discussion of System 1? No. All right, so what kind of system is System 1?
Anaerobic.
Good. It's anaerobic, right? It's not using oxygen, anaerobic. System 2 helps out the next step because we're only up to around 20 seconds or so worth of energy, and most of us have to move for more than 20 seconds at a time. So we've got a bit of a problem if we run out of ATP after 20 seconds, right? If we can't make ATP, how do I get the shopping done? Just about pull the cork on my glass of red, on my bottle of red wine, but I can't actually pour the wine out and lift the glass. That's not very helpful, right? So what happens next? Okay. Next step is a system called glycolysis. And you might notice that glycolysis looks a little bit like glycogen. And that's because not quite as much as glucose, but that's because it's a carbohydrate system. Right? Glycolysis is responsible for the next phase, and we need some food substrate now. And we said on Monday, that carbohydrate is a much smaller molecule, and it's easier to break down, so we can get at it quickly, any energy that's stored in carbohydrates. So System 2 gives us the ability to get at some energy bonds stored in carbohydrates. And system 2 is a little bit daunting when you look at the main diagram. Okay? So, I've spent many hours of my life learning glycolysis step by step, every single long, complicated word, and I don't know it. So we're not going to go there because it's a waste of time apparently trying to learn it. All right? We're not biology specialists. We're not going to do cellular biology or clinical biology. Okay? We need to understand glycolysis so that we can use it as PE teachers and coaches. Okay? So, we don't need most of the bigger words here. We do need the overall pattern, and we need a couple of the bigger words. Because we have to understand the overall pattern to see how we use it in training, and then how it feeds into system number 3. So, glycolysis has 10 steps, and it occurs within the muscle cell in the main part of the cell, as does ATP-PC, because that's where the ATP and the PC are floating around. This diagram is in your books, so don't worry that you can't see the words, because it's on, I think, page 35, somewhere around there. And we start off with a carbohydrate molecule. Carbohydrates, remember, have 6 carbons in them. Whether it's a glucose or a glycogen, we're still looking at 6 carbons. Once they're broken down, right? Because the glycogen is going to break down to a glucose. So I have 6 carbons at the start of the system. Okay? And we have to break it down, we have to break it down, we have to break it down, and we end up with 2 3-carbon molecules just before halfway. And we have to break them down, we have to break them down, we have to break them down, and we end up with 2 3-carbon molecules called pyruvate at the bottom of the system. Now, where does the carbohydrate come from? All right, it can come from the bloodstream because we have glucose in our bloodstream. And we can transfer it into the muscle tissue, and it can be used in glycolysis. If I use glucose, I come in at the top of the system, I feed it all the way down, I end up with pyruvate. If I use stored glycogen, because we store carbohydrate as glycogen, either in the muscle cell or in the liver, right? If what I feed into the system is glycogen, then it comes in 1 step down, breaks down, breaks down, breaks down, breaks down. I end up with 2 molecules of pyruvate. So the only difference between the glycogen and the glucose is the glucose comes in 1 step higher up. But that 1 step makes quite a big difference. Okay. The goal is to create ATP. Right? That's what we want to do. We want to make energy that we can use for muscle contraction. That's the whole point of the system. And what we see is not very much ATP. Right? So we really haven't created very much ATP here because we're only going for 20 seconds. When we break down carbohydrate, it's very useful because we can do it quickly. But we also don't get tons and tons of ATP. If I use this color, can you see it? Okay. So, I put in glucose. and I end up with pyruvate. Okay? I make 2 ATPs in this side of the chain, and I make 2 ATPs on this side of the chain. So for one piece of carbohydrate, I make 4 ATPs. But I have to use an ATP in this portion of the chain. And if I use glucose to feed the chain, I also have to use an ATP. So how good is your maths? How many ATPs do I end up with if I use glucose? 2. Right? So if I use glucose, I get 2 ATPs net. Right? I steal one, I make 4. Follow? If I use glycogen has to go through an extra little breakdown to get into the system. But it means it misses that first steal. So, how many ATPs do I end up with from a glycogen? 3, right? Use your fingers. 1, 2, 3. All right? Either way, this is not a lot of ATP. Right? Now, we're only talking about one molecule of glucose, and obviously we've got a lot more than one molecule of glucose or glycogen available to us. So we can make a bit more than 2 or 3 ATPs at any one go, but remember back to that initial chart when we looked at how many calories do we have stored as carbohydrate versus how many calories do we have stored as fats, right? Regardless, I've got a finite store of carbohydrate. There's only a limited store. So, given that I have a limited store and I don't make very much ATP from a molecule of carbohydrate, I can't make energy for a long time using carbohydrate. So system 2 runs from somewhere around 25 to 30 seconds. Bit of an overlap, and we've got some other things going on, to somewhere around 2 minutes. Depending upon the individual, how trained they are, what they ate last night. Having said that, glycolysis is important as an energy system. We use it a lot. There's a lot of times when we don't move consistently for more than 2 minutes. Right? If I walk out to my car and I drive to Walmart, then I don't work for more than 2 minutes getting to the car. I'm sitting down in the car, and then I have to start working again once I get to Walmart. Right? So I get a break in between. So we use glycolysis a lot. We use it a lot when we're playing sport. because quite often play doesn't last for very long, a couple of minutes or less. And it's the way we break down carbohydrate. So that means then, if I'm not a 100-meter sprinter or a 200-meter sprinter, because those are so quick I would be done on system 1. What if I'm a 400-meter sprinter or an 800-meter sprinter? What if I'm a basketball player or a football player? What am I using? Right? Often we'll use a combination, because you might be jogging down the pitch or the court and then have to sprint for the ball. So you might be using this and have to switch because you need more energy than is available through system 2. So when you're looking at this system, we've got a couple of big words in here that we need. But on the whole, I need to understand pyruvate. I need to understand the difference between glucose and glycogen. Okay. I want to know that I'm starting off with a 6-carbon molecule and I'm ending up with 2 3-carbon molecules. Okay. So we can simplify that ugly diagram quite a lot for our purposes, because all the clever chemistry stuff is fascinating, super clever. Right? I mean, it's amazing what goes on in a muscle cell when you walk across the bridge. But at the end of the day, as a PE teacher or coach, this is not the level you're working at. Right? So it's more information than you need, unless you want to dig into it and understand it better. So, same thing happens. If I'm training in a way that would use a lot of glycolysis to make the ATP I need, then I would see adaptations within that system. I'm going to see adaptations occur that help the system do its job better. And one of the things we see, because enzymes, remember, are very specific. So every time I break a molecule down, there's another enzyme that helps facilitate that breakdown. So there's lots of different enzymes involved in glycolysis. So we see some changes in those enzymes. Bless you. And we see an increased availability from glycolysis because we get more efficient at using it. Okay? If I train it properly, I can extend my 2 minutes to maybe 2¼ minutes. Again, 15 seconds, 20 seconds isn't a lot of time, but in sport, that 20 seconds can make a big difference, right? If I can get ahead of the other guy. The one I want you to think about, don't worry too much about these two, but phosphofructokinase is a limiting factor for this system. So we do want to pay attention. Unfortunately, it has way too many letters in it, but phosphofructokinase is one that we need to pay attention through. It's used at reaction number 3 in the chain. And if we don't have enough of it or it isn't functioning correctly, then it severely slows down glycolysis. And that means if it's slowed down, our ability to get ATP out of the system slows down. So when we look at people who do slightly longer sprints and more reps in their weight training, then we see changes to the enzymes that work in this system. We also see changes when we do endurance training. We'll come back to that next week, because did I mention anything yet about oxygen? So what kind of system is this? Anaerobic, right? Without oxygen, no oxygen. So we haven't got to talking about anything to do with endurance training yet. How much they change is a little unclear. When you look at the literature, one of the problems in XFIS is that people create their research in very different ways. The methodologies that they set up are very different. The populations they use are different. The kinds of training tools they apply are different. And so we have a range of different results depending upon the particular piece of research you're looking at. So trying to put an absolute number on how well this increases and who it increases for is difficult at the moment, because the research is not consistent. All right, so I can put glycogen into this system, right? And I store glycogen in muscle tissue. So another adaptation would be, that would be beneficial. Adaptations are always positive. Not always, mostly positive. Right? Certainly the adaptations we're looking for when we're training are positive. So if I have more carbohydrates stored in the muscle, that's positive. The more I have, the longer I can go. Yeah? Sensible, right? So if I train the glycolytic system, one of the adaptations we see is that levels of stored glycogen in the muscle go up for someone who's trained versus someone who's untrained. We'll come back to the endurance training next week. We don't see quite as much change if we're doing short, sharp sprints, short, sharp lifts. As I say, the research isn't consistent because of the way they set it up. Now, when I make the ATP, one of the things with glycolysis, and this is where there's a big error in regular language. Right? One of the things we hear all the time is, oh, I'm sore because of lactic acid. I ran out of steam because my lactic acid built up. So, a lot of drop in performance, you know, I'm running, I'm slowing down, because lactic acid is built up. Lactic acid is not the major problem. The major problem is that during this process, we give off quite a lot of hydrogen ions. Right? Hydrogen ions are acidic. All my enzymes at every step don't like a change in acidity. They like their pH very stable. So as soon as I start piling in hydrogen ions into the cell and not getting rid of them, then the acidity goes up, the enzymes slow down, and I slow down making ATP. From the outside, that translates to someone slowing down in the race.
What about soreness?
We will come to soreness next week or week after. soreness is more to do with damage than it is to do with lactic acid. Lactic acid, we do get lactic acid out of the system, right? So pyruvate changes into lactic acid. It takes a hydrogen to do that, so it's one way we get rid of some hydrogen in the cell, and we create lactic acid. But lactic acid is highly, highly, highly unstable, does not stay lactic acid for very long. It almost immediately turns into lactate, and it's very easy to shift lactate out of the muscle cell and into the bloodstream. So, on the whole, lactic acid and lactate are far less to blame for the drop in performance than the hydrogen is, right? The hydrogen is the thing that causes the acidity more than the lactic acid or the lactate. Not that they don't play a role, but it's minor compared to the role of the hydrogen ions. Okay? Think it over. Because if you haven't got the picture, Monday is going to get complicated. Okay? So please make sure that you review it a little bit over the weekend. You know, during that great big long mid-break they had in the middle of the Super Bowl, when Katy Perry is going to be shaking her booty right? Try to divert your eyes away from the TV screen and look at your exercise physiology book. Okay?
I'm staring at you there.